If this were in real life situation, we have already saved 175 people's lives. So that is very everything.
Welcome to Stories from the NNI. This series features voices from the U.S. National Nanotechnology Initiative. I'm Lisa Friedersdorf, Director of the National Nanotechnology Coordination Office. I had an opportunity to speak with Evangeline Alicilla, a professor in the Biosystems and Agricultural Engineering Department and Director of the Nanobiosensors Lab at Michigan State University. Thank you so much for taking the time to chat with us this morning. Could you tell us a little bit about your work?
Okay, my research work is related to nanoparticles, specifically nanoparticle-based biosensors. And we use nanomaterials, and in this case, nanoparticles, as part of the transducing mechanism for rapid detection of infectious diseases. And especially we're trying to focus also on drug-resistant diseases. And we use magnetic nanoparticles, gold nanoparticles, and magnetic gold, which are proprietary to our lab. So the idea is to see how we can really address the needs of poor communities where potentially there are no or minimal electricity, minimal refrigeration, minimal skills, and so on. My realization, I used to have fancy designs, and my realization was when I went to Mexico in the highlands, and we got a small project or budget from Kellogg to test our biosensors for tuberculosis, for rapidly detecting tuberculosis. Just the background of tuberculosis, there's 1.5 million people dying every year. 1/3 of the global population is infected, but without symptoms probably. It's basically a disease of among majority of the populations are really poor. And so we got this funding, and then when I get to the highlands, I realized that many of our designs do not actually fit into the system because there was hardly any electricity, there was no refrigeration, and so on. That's when we came back, and I thought there has to be some ways of using cutting-edge technology, especially nanotechnology, because that's where my area is, and addressing the needs of the poor. And so that's how we developed these biosensors, nanomaterials that has potential impact. So right now, for example, the conventional method of detecting tuberculosis is the smear microscopy, which has an average sensitivity of maybe 50%, 50 to 60% on average, with a specificity of about 90%, 85 to 90%. And we thought, well, if we can improve that method using nanotechnology, they already have the infrastructure in the rural areas. Maybe we could take advantage of that. And there was a paper that showed that if we can improve the TB detection from this current 50% to 87%, and from the current 85% to 97%, we could expect to save about 400,000 lives a year. That became our target. And so right now we have developed that technology, and our results from Mexico, India, and Peru, we have shown that the use of magnetic nanoparticles in the smear microscopy, so we call it nanoparticle assisted smear microscopy, we have moved from the data set show that the conventional method, we had 65% sensitivity. With the magnetic nanoparticle, we were able to go to 100% sensitivity. And for specificity, the convention gave us 93 to 95% specificity. With the magnetic nanoparticle, we were able to go into 100% specificity. With 100% sensitivity and 100% specificity, we can go back to the paper that said, yes, we could potentially save thousands of lives. And in this case, if this were in real life situation, we have already saved 175 people's lives. So that is very everything.
You've given us a great background on what inspired you to get into this work and the impact that it's already having. You mentioned the rapid detection. So is the direction you're taking is being able to identify this disease early so that people can get treatment, and that also impacts the spread of this disease?
That's correct. So the magnetic nanoparticle-based system gives us result in about 20 minutes.
Okay.
Whereas if you go into culture, we're talking about tuberculosis here. If you go into a culture, you require at least like 6 weeks.
Wow. And in the meantime, there's possibility of spread from the contaminated person.
Yes, definitely. I mean, TB is a highly infectious disease. So according to some literature, for every one person that is not detected in time, that person infects 10 others before he gets diagnosed and treated.
So this type of rapid detection in the field is really important to handling this disease.
Absolutely. Absolutely.
The technology that you're developing using these magnetic nanoparticles and smear microscopy, is this applicable to other diseases beyond tuberculosis?
Yes, it is very applicable. It's a platform technology. The idea is the magnetic nanoparticle captures the bacteria or a virus in this case from the clinical sample, and then it concentrates it. And so now you have a higher number. That's why the tuberculosis case, where that conventional microscopy requires 10,000 cells to be able to see it in order to detect it. Now with the magnetic nanoparticle, because it's extracting and concentrating, we can now see the disease with even 100 cells. You see the difference? That's why the magnetic nanoparticle improves the sensitivity, because it captures it and concentrates it. And the same case with dengue. Dengue affects like almost half of the world's population is at risk of dengue. The same case with antibiotic-resistant organisms, in a way that will make it fast for medical people to get a diagnosis and implement treatment as soon as possible. So that's the idea of extracting it, concentrating it, and then we use also our gold nanoparticle to detect it as well, so that then the diagnosis can be done from days to weeks to just maybe 20 minutes or an hour. And for example, for the smear microscopy, we're only adding at most 5 cents to the process.
Could you talk a little bit about how you have interacted with the National Nanotechnology Initiative?
When in 2003, I was invited to be part of the development of the roadmap for nanotechnology for food and agriculture. So that was my first real exposure to nanotechnology initiative. I have always been part of review panels for centers and so on. And I really saw the value of nanotechnology, and this is where I have organized a global alliance for rapid diagnostics, and it's based on nanotechnology-based diagnostics, and we are now in 17 countries, and all these people are very interested to understand and know how we can have a breakthrough in diagnostics and treatment using nanotechnology. Just last month, I organized a workshop, a one-day workshop on nanotechnology and biosensors in Peru. I'm trying to do this on my own way of doing things, and we hope to do this again in Nepal. And then in the Philippines, I was in the Philippines in August. I did the same. I had like almost a day of a workshop and lectures on nanotechnology and biosensors and how they can be useful in their area of research as well as in their diagnostic development.
Well, that's really great to hear. I'm definitely going to look for more information on your global alliance. That sounds very interesting. I wanted to let you know that we facilitate conversations among researchers in a variety of areas, including environmental health and safety aspects of nanotechnology. And those conversations are really expanding into areas like nanomanufacturing and nanomedicine. There will be more information soon on nano.gov. of, but I think that what you're doing and certainly the global interactions that you've had, it would be very useful to have you engaged in that discussion. Well, is there anything else that you would like our listeners to know?
One thing that I would really like to encourage listeners is that there is a lot to gain when you collaborate globally. And so that's why I've been so excited about this Global Alliance for Rapid Diagnostics, because it helps you know what we call the clock speed dilemma, because innovation can occur in many parts of the world, and research hypothesis testing can actually occur simultaneously from different parts of the world. So we just, for example, with our magnetic nanoparticles that we share with other collaborators, and I said these magnetic nanoparticles and gold nanoparticles are currently in 17 countries. People there would use it for something that we would not have thought of. And so there are these innovative ideas and innovative different people look at it from different perspectives, and then they'll say, well, I can do that, I can do this, and we were like, wow, I would not have thought about that. This clock speed dilemma for research is definitely one advantage for global collaboration.
I really appreciate you taking the time to tell us more about your research and the impact that this application can have. I think it's a really important story to tell.
Thank you, Liz. I really appreciate that too.
Thank you for listening to this episode of Stories from the NNI. If you would like to learn more about nanotechnology, please visit www.nano.gov and check back here for more stories.